Listen, guys, I've got to know what I'm looking for here. I know we're running out of time. We're all running out of time, but how am I supposed to? No one has found it.
What do you see? Come in. Can you hear me?
Normally this would be a pretty bad day. Might even be my last day, but I'm going to tell you why this, at least what it represents, might be the best thing to happen to any of us. See, this is a DNA shredding, cancer killing bomb. Now, before I explain any of that to you, I've got to explain what's going on. I've got to catch you up on some pretty complex science. See, we're talking about CRISPR systems. It's a bacterial immune response. It's kind of like our immune response. Stands for clustered, regularly interspaced, short palindromic repeats. And that's a lot, but here's the best way I can explain it. In a bacteria, there are these sequences of DNA, and they're all regularly interspaced, and they're all palindromes, which means they can be read the same front to back as back to front. Think mom, or wow, or race car. In between these sequences of DNA are other sequences of DNA that are from viruses that the bacteria has fought in the past. See, when it kills a virus, it takes a piece of its genome to remember it by, so that whenever it sees it again, it can identify it and destroy it.
There's a lot of different CRISPR systems out there, and these CRISPR systems, they operate mainly by detecting when a virus is there by using an RNA guide. They then use these effector proteins to get rid of the virus, essentially.
Those proteins are called Cas proteins, or CRISPR-associated proteins, and there's some famous ones out there like Cas9, which is often used for gene editing, and Cas13, which is used for RNA editing. But in 2023, Ryan Jackson and his lab of USU researchers discovered a unique Cas protein called Cas12a2.
Cas12a2 is unique from other CRISPR systems in a lot of ways. When the complementary RNA binds, it opens up a catalytic site that recognizes any DNA sequence and then cuts it. And it does this over and over and over again. We would call this multi-turnover.
So here's how 12a2 works. You give it an RNA guide, a genetic code. 65, what is it? What is it?
8675309.
And it'll look for its exact pair. And when it has its exact pair, it'll activate and start shredding DNA, effectively killing the cell, blowing it up. But if it's even one number off, it'll do nothing, leaving healthy cells intact.
Now we have a tool that could go into both a healthy cell and an unhealthy cell, and because we programmed it to kill the unhealthy cell, and what I mean is it's cancer, it'll kill the cancer cell and leave the healthy tissue alone.
Can you believe that only just now are you caught up to where we were in 2023 when I made this video?
We discovered a system, we're calling it Cas12a2, that targets RNA.
Now you might be asking yourself, well, why are we talking about what we've already discovered then? It's because at the time we discovered it, we weren't really sure that it could do all that we just talked about it being able to do. We had to be sure, and now we are. And here's how. See, the researchers just published this new data, and you're probably thinking, whoa, this is a lot. But let's just focus in on this here. Imagine that the green in this graph represents cancer cells, while the red represents healthy ones. As you can see, a few days after just one dose, the green cells are all but gone, showcasing that Cas12a2 is able to kill targeted cells and leave healthy ones intact.
There's still more that we need to determine with Cas12a2 and its mechanism, but as far as applications, the next step is really narrowing the gap on delivery. If we're going to use these systems systemically in humans or animals, there really needs to be some type of way of delivering the system where we can have it target specific cells in the right dosage.
Basically what Kadin is saying is there's a lot of work to be done yet if we are going to use this in humans someday. But this is still a major milestone and a pretty significant breakthrough in medical science.
This is basically adding another tool to the therapeutic toolkit for cancer. It's a way to advance all the genome editing that's already happening with CRISPR. This could be a huge game changer for science, medicine, and agriculture.